Our group members for this Journal Club presentation are Calvin Beardslee, Nancy Pearson, and myself, Nathan Moffat. We broke down and analyzed Figure 4 from the article titled A VTA to Basal Amygdala Dopamine Projection Contributes Signal Salient Somatosensory Events During Fear Learning. Next. The big question in this article was about fear learning and its applications in the natural world. Fear learning is where a certain stimulus is associated with a fear response, or in this case, an auditory cue is followed by a foot shock, which then triggers a defense mechanism, which could be a fight or flight response. Fear learning is associated with anxiety disorders such as PTSD and anxiety, where fight or flight responses are stuck on. So understanding the mechanisms behind why and how these disorders are caused can help improve treatment options and give people living with these disorders a better life. Next. So we learned from the previous presentation that BA projectors are activated during CS to US pairing. We also learned that most of the BA projectors are dopamine neurons. So there's an increase of the BA projectors in response to the foot shocks. Therefore, if dopamine neurons in the VTA and BA projections contribute to formation of auditory cued fear-based memories, then inhibiting them would limit fear learning and create a lesser reaction to the fear stimuli. Next.
So the main question for figure 4 is, will the inhibition of VTA dopamine neurons during fear learning decrease the fear response when later triggered? And as depicted by this cartoon, so if there are no VTA dopamine neurons active during the auditory cued foot shock, will this cause less of a fear memory formation? And in turn, what effect will this have on the mouse when it's later stimulated? So will this mouse, when it hears the auditory cue, freeze, or will it not freeze? And so will it be a brave Jerry or will it be a scared Jerry? Next. The general approach for this experiment included a light-activated viral dopamine inhibitor that was injected into the VTA of the experimental mouse group. And in the GFP group, which is the control group, they were not injected with the dopamine inhibitor. A optic fiber was then implanted over the injection site. The experiment was conducted, and during the foot shock, the yellow light was shined on the VTA to inhibit the dopamine in the VTA. The freezing levels were recorded, and then post hoc analysis of immunohistochemistry was conducted. And so a lower freeze rate, as stated previously, would indicate a braver Jerry, and a higher freeze rate will indicate a more scared Jerry. Next. So for figure 4, part A, this is a schematic drawing that depicts the light-activated dopamine inhibitor being injected into the ventral tegmental area in the experimental group. And this inhibitor is activated by a yellow light, which is depicted in part B. And this figure depicts the initial expression electrophysiology, and it shows on the top the holding potential and on the bottom the potential response. And it shows an inverse relationship between the current and the hyperpolarization. And so this yellow light is going to cause the inhibition of dopamine neurons produced in the VTA through this strong hyperpolarization and the robust outward current, which is going to change the cell membrane's potential and make it negative, which then makes the neuron unable to generate action potentials, and then in turn transmit these signals outward. And in C, we see demonstrated on the left the experimental current, and on the right the experimental hyperpolarization. and these values quantified, and this is correlating to the inhibition of the VTA dopamine neurons, and it depicts the peak and the steady state where the conditions have reached a steady state after being excited for some time. Next.
In figure 4, part D, it shows the schematic for parts E through H. We can see the light-activated inhibitor is injected into the VTA of the DAT Cree mice. We can also see the optic fiber implanted above the injection site and also the yellow light that will be applied. Figure E shows an image of the control mouse's, or of one of the control mouse's brains. The white dashed line in the upper right corner is the placement of the optic fiber. The scale in the bottom right corner is equivalent to 200 nanometers. In this image, there is a mix of red, green, and yellow. The red represents the stained tyrosine hydroxylase, which is an enzyme that makes dopamine. The green is the VTA dopamine neurons, and the yellow is where they both overlap. Figure 4 part F is the schematic of the yellow light In figure 4 part F, it shows the schematic of the yellow light illumination during the fear conditioning protocol. There are many lines in the bottom of the figure, and in between these lines is a 1-second period. You can see towards the bottom right of the figure that the light starts 1 second before the foot shock and lasts 1 second after the foot shock. So in total, the light lasts for 3 seconds. The electrical foot shocks last for 1 second, and right before the shock is applied, there's an auditory cue, which is a tone that beeps for 0.1 second at a frequency of 1 hertz. Next.
All right, for figure 4G, it shows the trends between the experimental and control groups over 4 days of the behavioral experience. So starting with day 1, it starts pretty much the same, but like I'd say the experimental group is noticeably slightly lower. And for day 2, it's still a little bit lower, but lower about the same as the control group. In day 3, you see like it's a lot different. It's a control group has a much higher freeze rate, and whereas the experimental group has a much lower one, which is a result of the foot shock driven activity of the VTA dopamine neurons on training day, like contributed immensely to the formation of auditory cued fear memory. And because the experimental group did not have that, because they had an inhibitor on their VTA dopamine neuron, that you see this drastic change in between both the control and the experiment. And also on the 4th day, the contextual fear retrieval was tested and found to be relatively similar as well. And for figure 4H, this is the data taken between the freeze rate of 6 mice in each control group over the same 3-day behavioral experiment. And it's noteworthy to say that one mouse in the control group was observed to have an unusually low freeze rate and was considered an outlier, as it was at around 26% compared to the rest of the data points. And overall, the data suggests that the experimental group's suppression of the VTA dopamine neurons had a very significant effect on the fear memory response as compared to the control, who had the same, like uninhibited VTA dopamine neurons. So you can see there's scared Tom, or scared Jerry, and strong Jerry, like the control, and then the experimental group.
So in conclusion, for figure 4, the dopamine neurons in the VTA do play a part in auditory cued fear learning, fear memory, and fear response. Specifically, that inhibiting the dopamine neurons in the VTA will reduce fear response when later stimulated by auditory cues. So referring back to the cartoon, if there is no VTA dopamine neurons stimulated during the auditory cued foot shock, then later when the mouse is stimulated by the auditory cue, it will be brave Jerry and it will have less of a freezing response as the control group would.
And lastly, for our criticisms, we found that part C, G, and H had the same black and red colors consistent with the experimental control groups, but here they meant completely different things, which we found was very confusing. And we also, as I recently stated in figure H, one control group mouse had an unusually low freezing rate, becoming an outlier within the data and not being included. And lastly, our sample sizes were very small, so it's difficult to determine the certainty of the results and also the importance of them as well. Lastly, we have our references.